what is the IGVF and what is its primary objective? Well, the IGVF is a research consortium supported by the National Human Genome Research Institute of the NIH. It actually consists of 26 different awards that are distributed across 60 investigators, more than 60 investigators at over 40 different institutions. The primary objective of the IGVF is to advance our understanding of how genetic variants impact molecular and cellular systems to cause diseases. Sequencing technologies have made it possible to systematically catalog genetic variants in the human genome, and population studies have been able to link some of these variants to diseases, but we don't understand how the variants are causing disease. And that level of understanding is really critical for being able to accurately project people's risk of having diseases, to understand if a particular variant is causing a disease that somebody has, or to try to develop new drugs that might help people with those diseases. So what IGVF does is it brings together experts from many different fields to use state-of-the-art single-cell technologies in order to really go deep on studying genetic variants and how they're impacting different functional elements of the genome. In addition to studying this in a broad array of cellular contexts, this is all being brought together in a public-facing catalog to make it possible for investigators all across the field to use the types of data and tools that we're developing to further investigate genetic variation and its impacts on the genome.
The NIH Consortium for the Impact of Genetic Variation and Function is making incredible advances towards our understanding of human health and disease. The fields of genetics and genomics are not new, and in fact, almost 25 years ago, we had the first announcement of the draft sequence of the human genome. But that was really just telling us what the letters are in this book of the human genome. And if we continue with that analogy of the genome as a book, once we knew what the 3 billion letters were that made up the human genome, we've spent most of the last 2 decades understand how those letters are arranged into words and sentences that make up the text of our genome, and how those words and letters change across different genetic backgrounds and different individuals. But what we've been missing this entire time is the dictionary to tell us what each of those words actually mean. And it's that function of those words that the IGVF Consortium is working towards understanding. You can imagine that if we don't know what the meanings of the words are that make up the text of our genome, it shouldn't be that surprising that we have such challenges with treating some of our worst and most common diseases that have genetic contributions, whether it be neuropsychiatric, neurodegenerative conditions, autoimmune disease, cardiovascular disorders, or cancer. And so really understanding this function and this dictionary that makes up what each of the words in our genome is doing and how that varies across cell types, stages of human development, and in response to our environment and different pharmacologic perturbations is really going to be critical to making those next advances towards understanding how our genetics and our genome contributes to human health and disease. As a recent example of some of the work of the consortium in understanding how our genome contributes to human health and disease, we wanted to explore how the environment of a cell or the mechanical properties around a tissue affect our genome and how that might be involved in disease progression. The mechanics of a tissue are critically important in numerous human disorders, thinking about things like tumor development in cancer or fibrosis, or even things like stroke or neurodegeneration, and even aging. All of these are diseases or processes in which the mechanics of our tissues change over time. We know that that converges on our cells in their genome to change in gene expressions, but we didn't really understand how and why that takes place, where that happens in our genome, and how those changes in genome structure affect the genes and the levels of those genes that are involved in disease progression. So we put cells in different environments of different mechanical properties and then observed thousands of changes in levels of certain genes and tens of thousands of changes across the genome in the structure of different genomic elements. We then mapped many of those to their target genes that we could then link back to genes and regions of our genome that are associated with complex human diseases like fibrosis. And so we hope that this very detailed mechanistic understanding of how our genome responds to environment and how that is involved in complex disease processes like fibrosis will help lead to new drug targets and new druggable mechanisms in areas of human disease that have otherwise been recalcitrant to many of the existing approaches and modalities like fibrosis.
I was incredibly lucky to join the consortium as a new graduate student, and then immediately surrounded by a bunch of people that were interested in the same things as me, but also had a lot of different forms of expertise that I could lean on and learn from. Also really nice to be able to talk to other graduate students about their journey, not just at my university, but also across the country, really, and even a little bit of beyond borders. So all of this collective expertise really helped me learn and grow in various ways because people brought in different specializations. Like there are people that knew about different experimental techniques, statistics, machine learning, and even perspectives from the medical side, letting me know what it's like to receive, be on the receiving end of medical care based on this kind of research. What's really great is that all of this knowledge and data exchange is far larger and greater than any one lab or any university can achieve on its own. And the really nice thing is that you don't have to reinvent the wheel every time we want to do something. If I was ever stuck in my research, I was able to get feedback from dozens of different people that could attack my research from different angles and make sure that I was doing the most rigorous work possible. You know, saying like, have you thought of this? And like, tell me why you did it this way. And you know, always just like knowing that we're just here for the same goal in like a very non-competitive way.
What excites me most about IGVF research? Well, our DNA plays a major role in determining our risk for diseases, from type 2 diabetes to heart disease to Alzheimer's disease. Fortunately, our DNA also holds the clues that we need to cure these diseases. In IGVF, scientists from around the country have come together to understand how our DNA throughout our genome affects our genes and cells to lead to disease. And by better understanding this process, we can design new specific therapeutics that target the root causes of disease, make sure that each person is taking the right drugs at the right time, and transform our understanding and ability to diagnose rare diseases. One example is in the space of congenital heart defects, which affect 1% of children. And defects in the heart are in large part due to defects in our DNA. What we're trying to do is identify all of those DNA changes, DNA variants in our genome and in the genomes of kids who are born with heart defects, and learn how those variants disrupted the very early process of heart development to lead to disease. By understanding this, we might be able to design new types of genome therapies that go in and perhaps correct the DNA and guide heart development in a productive way. And so by better understanding this link between our DNA and disease, we can design new types of drugs that target these processes.
Hi everyone, I'm Jishnu Das. I'm an assistant professor at the University of Pittsburgh, and I wanted to talk to you about how IGVF has helped my career as well as my own research program. So I started my lab in January 2020, and I was very fortunate to receive this IGVF grant in the second year of my lab in 2021, and we embarked on this journey to really use networks, biological networks, to understand the impact of genetic variation on function. So I'll tell you a little bit about two aspects. One, how this fits with my research program, how it has enhanced my research program, and the other aspect of how it has had a transformative impact on my career. So I run a computational systems immunology lab where we use machine learning and network approaches to study immunomodulation, What that means is we harness the power of artificial intelligence and machine learning to really understand how you can modulate your immune system. And now one key aspect of what modulates your immune system is genetic variation. And as the acronym IGVF stands for, the impact of genetic variation on function. So what we're really doing is we are trying to model the impact of genetic variation on these complex cellular and molecular networks, specifically within our center, where I'm co-leading this with Harinder Singh's lab. We are looking at a specific kind of immune cells called B cells. These are very important in the adaptive immune response. What we are doing is we are constructing these high-resolution cell state specific networks that we call GRNs, or gene regulatory networks, and how the impact of genetic variation affects these networks. This has very wide-ranging implications, from chronic autoimmune diseases to studying vaccine-induced responses. And this is a core part of what my lab does. So the IGVF mission and what my lab does are highly synergistic. That's the research part. But how has it helped or transformed my career? As an early career PI, as a junior assistant professor, I was really fortunate to receive this fantastic award. And in conjunction with Harinder Singh's lab, we really tried to take our research programs to new heights. While the proposed work was both central to what we were interested in, this has helped propel my lab and grow my lab, and more importantly, interact with this amazing IGVF community. A critical part of both growth for me and members in my lab that are part of this IGVF team are the IGVF community. And graduate students, postdocs, staff scientists in my group have really benefited from interacting with this IGVF community, which is amazing, creative, and has a lot of great ideas. So these interactions have propelled not only my career, but also the career of people in my lab, and that is why this community is so awesome. So this is really a place where you can do amazing science, good research, but also further your careers, and that is why IGVF is awesome.
I think it's crucial to build a comprehensive catalog of variant effects so that scientists can have a resource that they trust and can go to see whether their variant or gene of interest impacts different tissues or cell types differently, and using various experimental assays. I'm a neuroscientist by training. I just got into neurogenetics during my PhD, and I know a lot of my neuroscience colleagues would really appreciate having a resource where if they have a gene of interest, they can go to it, see how it impacts the brain and other cell types or tissue types, and really get an answer by seeing the different experimental methods. One place to see everything and not have to rely on AI or ChatGPT.
What are the key problems in the field that IGVF is addressing? So the Human Genome Project gave us the sequence, and ENCODE gave us the parts list for the human genome. But now we need to understand the impact of any sequence change or genetic variant on human health. And as a field, we're actually pretty terrible at guessing what those are. And this has caused all sorts of problems for genetic medicine. An example of this is when genetic testing identifies variants of uncertain significance. A variant of uncertain significance means that the person really didn't get a definitive diagnosis for whether or not they're at risk for disease, and their doctor can't give them the optimal care. And these results are also a barrier to the kind of genome-guided precision medicine that was imagined around the turn of the century, when we knew what the sequence of the genome was going to be. As part of IGVF, we have shown that functional data and variant effect predictions can overcome this barrier. And our goal through IGVF is to make variants of uncertain significance a thing of the past and make genomic medicine more impactful for all.
What's exciting about the IGVF catalog is that it's an entirely new way of representing the information. So whereas previously we were able to annotate the human genome and talk about the regions that are active in different cells or in different conditions, this new type of data perturbing the human genome, it's really represented instead as a network, because a perturbation of a part of the human genome could have different effects on different parts of cells. So we measure from the variant to the element in the human genome to genes and downstream cellular phenotypes, or even human phenotypes like disease.